So last time we said that once we get the raw data from a sequencer, whether the next generation sequencer or third generation sequencer, we get what we call short read or long read. We have to do quality control first to make sure that the quality of the reads are in good shape, and after that we can move um, to the next step. The next step in most of the next generation sequencing applications is read mapping. And in the read mapping also we discussed all the steps of read mapping, beginning from uh, downloading the sequence of reference genomic FASTA format for that specific organism. And then we need to index it both using SAM tools and Mapper. There are different mapper or aligners. And then we will align the reads in FASTA file to the reference genome, and we will get SAM file. Then we can convert, as I give you in this assignment 7, to complete that by converting the SAM file into BAM file, and then sort it or sort the aligned reads and index them. Then we will be ready for the next step. Okay. And also it says that the uh, SAM file or BAM file include many information. We discussed how to manipulate that, how to get some information, and I hope that if you want to go further, in bioinformatics, you need to know more about how to manipulate and how to get information from a map or some file and how to filter it. Okay. But it's still the BAM file, while using it is the downstream analysis, okay, as we will see now. Okay. So today we'll talk about genome assembly. And genome assembly, that means if we get the short read from sequencers, the sequencing strategy itself is the whole genome sequencing. Does that mean we are getting sample from any organism, and our purpose will be to sequence its genome? That means the entire genome. And we call that all genome sequencing. That means we are not focusing on a specific region, but we take the samples and the DNA sample from the organism, and we follow the strategy to sequence all genome. That means we'll get, at the end, we will get short read that depend on which protocol we are follow, which platform we are used. The reads length may be 100, maybe 20, 200, maybe 300, maybe 400. That it depend on the sequencer and the protocol that we follow. But we will get the reads, and then we follow all that step until the point of read mapping that we discussed last time. Okay, and I gave you like 2 assignments. So from read mapping to a reference genome, then we will continue assemble the genome, and we'll show you how to do that in a different way. The assembly genome is, we use it in many applications, of course, like if you need to identify an organism. And also it is important to know the sequence of genome of any organism, right? Because we need to know the different gene and if there are any variant by variant, like any mutation, um, single variant mutations or SNP, single nucleotide polymorphism or deletion or insertion or structural variation or any kind of um, mutation in the gene. So it is very important to know it, and the only way to know that is to assemble the genome, right? And we use it in different aspects, like to identify viruses, to identify bacteria, And even for human, we use it also to identify, to know like the relationship, between the two, at least, to learn the different genetic disease in human, and that will help in treatment, diagnosis, treatment, and also it will help in drug discovery as well. So genome assembly is really very important, and we use it all the time in different, um, for example, like the outbreak of Ebola virus, for example. So we need to sequence all genome to know which variant, and also SARS-CoV-2 or COVID-19 viruses. Um, Also, we need to know like which variant. Already we know there are many variants of this virus, and any variant has um, different symptom, in the patient, right? Some is very infectious, and can spread very fast, and some very slow in spreading, and pathogenicity also is not like, very severe. So we need to know this kind of stuff. So to do that, we need to sequence the genome. When we come to sequence the genome, there are two strategies. One is a reference-guided genome assembly. And this, if we know the genome, if already we know the organism, that means the organism has a reference genome, as we uh, discussed, we said that we have the databases, and many governments like United States of America invested too much money in developing this database. And this database include reference genome for all known species or organism. So if we already know the organism, then we can use the strategy of a reference guided genome assembly. Okay, that means already we know the sequence. But in some cases, maybe um, uh, we don't know the organism itself, the species. We have the DNA, or maybe the organism itself, it doesn't have a reference genome in the database. So in both cases, then we need to think um, about different strategy, and that different strategy we call it de novo genome assembly. De novo, that means something from scratch, something new. Okay. And here we do not depend on a reference genome. Okay. Even sometimes, if we know the reference genome, in some study, we use also de novo genome assembly, especially if we need to avoid any bias from the reference. And we need to discover new gene, for example, or new marker or something like that, and we need to be like completely independent from any reference genome. Then still we can use de novo. And there are different ways to do it, but most of the time we are using a reference guided genome. And this when already we know the reference genome. So in this diagram, it will show that um, those are the reads. So there are different kind of reads. This is the read that we get in FASTQ file from the sequencer. Then here we have two options, either to go to reference guided or reference based assembly. And here we have to use a reference to align the reads. And then we get what we call consensus sequence. Because if you see here, from when we align the sequence to the genome, that means each one of these reads um, will be placed on the region of the reference genome. So already we know that the different organism algorithm we discussed last time about finding the location of read. So once they are aligned like this, they can form what we call it consensus sequence. That means because there will be overlap, as we visualized in using integrated genome viewer last time, IGV. You show how the reads can be overlapped. From that overlapping, we can get what we call consensus. Consensus, that means when we aligned in a column or any base, uh, we choose the agreed base. So we say, okay, so this is a base. So at the end, we got, we call it consensus sequence, and that will be the genome. That means this genome, it will integrate. It will not be exactly like the reference genome, right? Because here what we do, and as we visualized last time, there will be a column, for example, like base or nucleotides, mutation or anything different. So in this case, um, we will integrate, we will use that whatever, mutation. If they agreed in a specific base, then we will use that base. We're not going to use the reference sequence. Right? If they agreed with the reference base, then we use that base, and so on. So it's a matter of just knowing the variants and integrate the variants in the consensus, and so on. In case of de novo, also we have to use different algorithm, and we will discuss this algorithm maybe next time, maybe this time if we find time. And here we don't have any reference genome, but there is a specific algorithm that allow us to um, organize or to order this gene, this short read in a way that can create a consensus sequence. So this is the difference. See how this will be very easy, straightforward, because we have reference genome and we align the read, but here we don't have any reference genome. So we have to figure out, and there are many algorithms to do this, and we'll come and discuss them, and at the end we will get what we call consensus sequence. So these are the two strategies for genome simply. So now we'll talk, so we'll focus on reference guide or reference genome assembly. So already we know there are thousands of, thousands of, maybe millions of organisms now. But anyway, we can, just to be uh, realistic, there are thousands of organisms. Already we know their reference genomes, they are available in database. Okay. So whenever you come to to do any kind of genome assembly, then just go there and look in the database to look for that reference genome, and you know how to search for reference genome. And don't forget, always you can go to the nucleotides, ACBI nucleotide database, and you can use that search query. If you use refseq, then you can get that reference sequence, and you can type like whole genome or complete genome or something like that. So we will get the reference sequence. Okay. And the reference genome organism can be used in guideline for building. This is what we said, and we will integrate all kind of angel, that mean insertion and deletion, and single nucleotide variants. Okay, so don't confuse between single nucleotide variants and SNP or SNP, single nucleotide polymorphism. Okay, the difference is that, so when we discover, if you find any different from the different from the reference, like any base different from the reference, we will say we call that a maybe single nucleotide variant. That means it is a mutation, or it is a mutation, right? Or position mutation, single position mutation. But what is the difference between uh, single position mutation or single nucleotide variant and SNP? Do you have anyone has any answer for this? Because I think I explained this before in the introduction. Do you remember what SNP is and what is and and what single nucleotide variant is? Anyone volunteering? Okay, so single nucleotide polymorphism is single nucleotide variant. Okay, but the single nucleotide variant is not a single it's not a SNP. It's not a single nucleotide polymorphism. Okay, why? Because the definition of a SNP or a single nucleotide polymorphism only if the frequency of that single point mutation is at least 1% in the population. Then we can define it, then we can describe it as a SNP, and we can report it to the NCBI database. And the indi in the indi in and in the NCBI database, they will, if it is new, they will give it a reference number. Why? Because the SNP really has very important uh, role in many diseases or trait or sometimes as a marker. So this is the definition. So if I ask you in this exam, what is the SNP? Now you should answer. Okay, the question. Okay, I'm going to repeat this again. The single nucleotide polymorphism or SNP, when the frequency of that single nucleotide variance is at least 1%. That means if you have a population, whatever you know population, the study population, say, okay, we need to study like we have thousand of population, and that population say like of any country or of any race or and 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 after we analyze their genome, we found that it's a single nucleotide variant. So that means a one substitution. And when we compute that. Of course, some people some people they may have that variant, and some people they have, some people they don't have. But anyway, we find it, so we can determine the frequency. And you know what frequency mean, right? So just is count the number. And that sounds like how many variants, and after that we will bring the percentage, right? If it is at least 1%, then we can call that SNP or single nucleotide polymorphism. If it is not, then it is just single nucleotide variant. Okay. So this is the difference here between these. And also you should know what consensus sequence, and we discussed consensus sequence that as last time we display in the integrated variant, the integrated genome viewer, IGV. That when they overlapped, then you can get the consensus sequence, and then we use that consensus sequence as a genome, as the new genome. So for these, we will use PCF tools. There is a tools, we call it PCF, and we will use it. So we use for a simple, by the way, there are many tools, but here we will give you very light, uh, because really, here we have it's constrained here. So we are not able to cover like everything in details. So for example, if we need to cover all of this part, at least we need to cover all of it. Maybe we need like maybe um, 10 or 7 lecture just to talk about the variants and mutation and variant calling and annotations and many things. But anyway, still we will go and discuss some of these, and hopefully in the future we can read more about them. Okay. So this is like we disputed last time on how we get the consensus sequence, and I don't think that we need to explain this again because we talk about this many times. And it says read, they can be, and whenever like this, all of them, all read agreed, except here. So we consider this as error. So we call it the consensus sequence here will be instead of G. And if you see, it is different from the reference genome. This is the reference genome, and so on. So if we agree with the reference genome, then we like C, all of them C, then it will be C. But if our read of that individual, because all these read from a single individual, they are not from different individual, okay? Then we'll, why? Because they may be like mutation. So we call this mutation. So we're going to add this, and we call this consistent. Here you can see that the importance of coverage, because coverage will give you the how you the confidence, right? Because when you like, especially in science, when you reach any conclusion, always you have to tell me about how confident are you, right? So the confidence in statistics described by like p-value, for example, and described in different ways. So they are always, in science as general, there must be a matrix that measure how you can rely on this, because this is about science. There is nothing perfect, but it is just like approximation to the reality. And we'll try to reach to the fact, but still you cannot say what you get is a fact, right? So matrix here is really very important. And here what uses the coverage. And you see, for this, you can be very confident because here we have, see how many reads here we have. And because we have many reads cover this area, so we will be very confident that this area is really, there is nothing about it. But if you go here, for example, here A, see the depths, and we'll come and discuss this in the variant files. Here we have only one support this one, and this A it might come just by it might come by chance, right? Because you have a single, and here you have two, only two. So whenever the cover is not like too much, um, here the confidence will be low. And if you have very high coverage, then the confidence will be very high. We need to always, to remember this. And especially if you do like always refer to the coverage or the depths to be how confident that what you are doing is reliable. Okay. So it's all about something not to be reliable that depend on. And so you can use it in the downstream analysis. So this how, so at the end, because we need this sequence, consensus sequence, and this is what we call it a reference guided genome assembly. So here our goal is this sequence, and we call this a genome. So we talk about the variants and genotyping, and we don't need to talk about it before, but once you get the sequence, of course, you will think about what variant, what kind do you know about that variance, and that can be substitution, and also single nucleotide variants or SNP. We talk about the SNPs, insertion or deletion. And here I put the definition here. The SNP is at least 1% of the population. The frequency is supposed to be 1%. And of course, the SNP always have accession numbers in NCBI database. If you go to the NCBI database, DB SNP, you will find all the SNPs there. Okay. And if you are working in any population supposed to be large enough, like more than 100 person or 200, 600, 6,000, very large. If you make like a study like this and you discover some single nucleotide polymorphism and you find them in that population, you can see how you can see the frequency, right? The frequency, and from there you can maybe you can link them to any trait. For example, if they are a specific population, so they use this SNP to compare between different populations, like African and European and so on, and diverse people. They may have different SNPs that they can be compared and they can identify them, right? So this is very important. Also in diseases, and in some diseases like associated with Alzheimer, for example, we have apolyprotein E. This associated with Alzheimer. So genetically, if you so it's possible, just to sequence the genome and look for, and I think this in, if I can remember, this in chromosome 9. So if you go to that position, if you sequence, genome, chromosome 9, or you go to that position specifically and see um, whether that there is specific SNP there. It's known. If you go and you find it, that means this is like risk factor that these guys will be affected by Alzheimer. Okay. The same thing also for breast cancer. And now this like mammogram, they are included. Some SNPs, they are there. So this is like an alert flag. Okay. That means, that women, it might be affected by breast cancer. Okay. Because the SNP or the SNPs driving, they call them driving SNPs, they are already there in the genome. And so that like, risk factor for to be affected by this kind of cancer. And the study now actually depend on finding these markers or um, uh, they call them driving, SNPs or variants or anything like that, and try to associate them to a clinical, because this is what is of course this is what is important, right? Nothing else. It doesn't matter, your color or your skin or your race. So what is important is your health, right? So when they map this to like disease or something like that, because that will make it easy to control that disease in very early stage. And if you are, for example, somebody at risk, uh, and the factor is there, then of course they can be controlled in any way. Now there are different ways to control this kind of. But anyway, the importance of these are really the importance of this one. So um, so here to know, we have to know what the variant code format. So already we have that BAM or SAM file, right? And that SAM and BAM file include the aligned sequence, the aligned read, right? So they are already aligned. They are aligned to the reference sequence, right? The second step in the genome assembly, what we need to identify the variation if there is any mutation. So we need to identify it. Okay, so this will be the second. And we call that by a process we call variant calling. Okay. And we will come and discuss about variant calling or variant discovery. But before we before we go there, just let's to understand the concept, okay, before we go and generate that file. But we use this to identify the variant, and by variant we mean mutation. There can be substitution, deletion, or insertion. Those are the common um, uh, mutation that will be discovered, and there is a program specific for that, algorithm also specific for that, and then we will create what we call variant call format file or VCF file. So now, so far, FASTA file, FASTQ file, BDB file, and also, FASTQ file is BAM and or SAM file. And this format again is really very important that any bioinformatician should know, because this is one of the basic things in any interview. If you've been asking about what VCF file and you didn't answer that question correctly, then you will be removed immediately, okay? Because they are not qualified to go any further. So this is one of the basic things that you should know. So once you get the BAM or some file, then you need to go to a different program, and that program will get a variant called file format. Okay, and we will discuss that because this format, this file is specifically include all variants, deletion, insertion, and substitution. Like the BAM file includes the aligned read. So if I ask you this is a major question that I'm going to ask in the exam, and now I'm telling you here. You should differentiate between the difference between BAM file and VCF file. In BAM file, that include the aligned read, the read aligned to a reference genome. That includes the alignment information. VCF includes the variant information. So how the reads that you get from your sample, they are different. They are different from the reference genome. So the different. All of them, they will be stored here. And a single file can include many samples. So many samples can be included in a single file format. Okay. So this file format include also from a two-part metadata or header section to hold the meta information, and a data section that hold the variant information. See, this is the same organization like BAM file, but here it will be different. And so when we talk about the meta or the header section, each line begins with this double hash. If you find double hash, that means this is a header. And of course, must be the first lines in VCF file. A metadata line describes the format and content of VCF file, and we come and give you some example. And also the understanding of this format really is very important. Again, like BAM file, and we say that's the beginning of by real bioinformatics, because included from there, like FASTA files that raw data, but any results of analysis, it will be the bioinformatics, because we are getting information from that file. Then BAM or some file, there are a lot of information that always you can get. The same thing also, VZF file, also you can get information from there. So the header file here include information about the header. We show you the first few lines describe file format, the file date, source program, and a reference used. So it might also include some information that which reference genome, because like human genome, they are about 5 or 6 version. we are using HG38, but they're like HG19, HG18, and so on. So they're different versions, right? So this, the reference genome, and by the way, if you are doing this, always stick to the same reference genome that you are using, okay? Because that, as I said, there are different reference genome beginning from HEG, I don't remember actually, it's in 2000, first one it was 2006, 2000, sorry, 2003, after that 2006, and 2000, um, maybe 19, something like that. So this is the latest one. But we are using HEG38 because this is stable and very well annotated. And now we have a new version. I think it's like HEG39 maybe. But it's still, it doesn't, it's not like it's good, of course. It may be like after like 1 year from now, maybe we'll switch to it. But now 38, this is the most stable one, very well annotated, and scientists did many effort to to associate, this genome or annotate it with different kind of annotation. So of course, the annotations I cannot just trash like that. So we are but it's still using that one. So you will find info and format, and you will see how info it will give you information about field, and format how the field that will be formatted. So this will be, the header will be like this. Like info and format, those are the most common ones. In format, you'll find like ID, the info, ID, number, data type, like for each field, and we'll come and show you some example here what these mean, okay? Like number, how many value, type, data type, is it integer or float or string, or most of the data type are integer, okay? And description of the field, number of sample. So this is the description of the field, name of sample, like how many sample in this, it will give you answer how many sample in your VCF file. And it is integer, and it is one value, okay? Again, DP, mean depth. Also, this is one of the important field that we have a single value. It is integer. Description is total depth. The depth that you talk about, because if you find a variant, for example, deletion, or say like substitution, for example, and depth is 1, it's not like if depth is 10 or 20 or 200 or 1,000. More depth, more confidence. Less depth, less confidence. And sometimes we need to filter based on the total depth. For example, if it's like depth is less than 5, then we need to remove that variant, okay, or not to consider it. And some example will show you. Also, the data format, the same thing also, like GT, this genotype. You would find description. This is a genotype, for example. Um, uh, this genotype quality, again, depth. You'll find also the format. So let's see how it look like. And so also, so after the header, you will find the data region. And in data region here, we have these 8 mandatory fields or columns, but also we have optional columns that will be added. And that optional columns, if you have multiple samples, um, then each sample will be added here with its unique number. If it's run number or run number and so on, and we show you in the example. But those are the fields like chromosomes, position, the position at which the variants or mutation or whatever, it is there, or mutation is there, the ID. If it is SNP, non ID, then it can, it's very easy to get from the database, right? It would be like from DB SNP database, and there will be accession number. If it is not, if it is not from available there, then there will be no any accession number. Ref, this is the base on nucleotides in the reference. And all this is alternative. That means which base is found in the genome. Okay. For example, if it is in reference, the nucleotide is A, what in alternative, what is the corresponding one? What's the aligned? Is it the same like A? If it is A, that's fine. Then we call it A. But if it is like G or anything else, that means this will be that mean mutation, right? So this is the difference. So this reference and this for alternative, that means and each one can be either A, C, G, or T. And here about the quality, and here filter also is very important. If you apply any filter to this file, just the filter will be written there, and also will show you some example, info and format will show you. So this is how I hope you can see this very clear here, and maybe we'll go also visualize some of these, the header, and these are the body or the data section, and each part of this include variant. So each line will be variants, okay? For example, here this chromosome number 20, and we say that the chromosome can be labeled in different way. Can be labeled as CHR1, and so on. Can be complete chromosomes, 1 chromosome, 2 chromosomes, and so on. It can be a number like this, but we should know that the first column here is the chromosome number. And this is the position in the reference genome. And the ID also, we talk about the ID, and we say that the ID, this is the reference SNP, and it should be in DB SNP, NC by DB SNP, if it is there. If it's not there, it will be like a dot here, like because there is no any reference, okay? Or there is no any SNP, okay? And then this is the reference. That means this is the base in the reference, and this is the alternative. And if you see here, in the original reference, we have G, but in our sample, we have A. That means here, we have a mutation here, right? And this is the quality of, same like quality we talked about before. And here, filter pass. Why? Because usually this column is supposed to be empty. When you align it first time, it will be completely empty unless you enforce any filter. Enforcing like the filter enforce here is the quality to remove any variant with the quality less than 10. So in this column will pass, that means the quality is more than 10. But if you find Q10, that means this didn't pass. So it didn't pass, they're not gonna type fail, but with the type the filter itself. The filter here, if you can see here, it's Q10. That means this is the like threshold or cutoff. If it is less than this, it will not be passed. So the filter will be written here. If it is pass, then there will be written pass. And also you can apply multiple filter, and they will be separated by semicolon. Okay, they'll be separated by because you may need to improve the quality of your VCF, and you apply multiple filters, and there are some command will show you how to filter, filter this but anything that they will be written here. And of course, after that, this is like information that we talk about, like here, the depths. Here, if you see DP, the depths. For this variant, we have 14. That means 14 read base. And this is not bad. And even you can filter this. Because if you see here, like this, we have 14. Also here we have variant because TA, that means they are varied. And the depth here is 11, and so on. But if you find the depth is very low, or you need to filter to keep only the variant with depth more than a specific cutoff, then you can use filter as well. And you can use any filter of this or anything of this, okay? So this is how information also included here, the depth filters, and they include this information. And now we come to the format, we come to the genotype. And in genotype, um, specifically, this is also really very important information. If you see here, we have three different samples, right? We have one, this is sample two, three. And here we have the format. So how the genotype formatted, and by genotype, it means, of course, um, the genotype of our genome, how it different from how it different from the reference genome. Okay, so this is the genotype or the allele. So later on we'll talk about the alleles and how. But if you see here the genotypes, this is the format how they look like. This is only the format, but they are right here. So this explanation to the enter here, like genotype, this is the genotype is 0, 1. For this sample, it is 0, type 0. Okay. For quality, for genotype quality is 48. And for the depths, it will be 8. And for HQ, we call this haplotype quality is 2 numbers. And again, if you go to genotype, here in the format, you will find the number is 1. If you go to each of these, the depths, you will find an integer. But if you go to haplotype quality, you maybe will find it 2. So this 2, that means it will consist of 2 numbers separated by colon. Okay. Again, here really there is a very important information. Let me to enlarge this a little bit so to understand what this means. 0, 0. This is the genotypes. Mean for these samples, because here we have 3 different samples, and this is the genotype for each of these samples. When we find 0, 0 in this sample, that means it doesn't, that means it is the first thing we'll come and talk about the alleles. And alleles that, as we explained before, that if you have like a chromosome, that part of the chromosome, we will get it from parent, one from mother and one from father, right? And after that, of course, they will be in the same chromosomes, and each gene will be in form of allele. Allele, that means it might be like different format. And the sequence of each um, allele, like each basis will be like, also we call it allele at the same time. So if you have a SNP here and here, also we call them alleles. Okay, because they do a specific thing here. So in this position, 14 something in the reference genome, they have this. And in the first samples, they don't have this. They don't have, because whenever you find 0, 0, that means it's the same like the reference genome, both of them, the same like the reference genome. And if the two alleles are similar, what we call that? Do you remember what we call that? Anyone from biology, animal science? What we call that? We call this homozygous. Okay. When the alleles are exactly similar, this we call this homozygous. And these each allele, so because there are two pair and post pair actually is the same. So here we don't have any mutation. But if you go to the sample number 2, and by the way, I say that the VCF file can take millions of samples. All of them, they can be in the same file, and each one will be given a column like this. Okay. And this is really very good about this file format, because it can include any number of variant, any number of samples. They can be in the same file format, and the analysis also, it will be very easy. Like bioinformatician, you should know how to analyze this, how to compare between these and these and these. This really, it's really a very big topic. But you should understand that, here, it's just like you can do any kind of comparison between different, assume that you have 100 samples here. Maybe you need to split your sample into control and not control, and try to compare, you know, between these two groups and so on, maybe three groups also at the same time. So all this kind of thing, um, it should be included in the analysis, and in the study as well, because they will give you an answer for different kind of study. For example, like if you need to compare, like, a group of two population, some like healthy people and diseased people. You want to see, compare. Also, you can divide them into two groups. But anyway, the sample will be um, oh, I'm sorry. I think I went too far. Yeah, so this just let me finish this. So for example, here the first allele actually is G, but instead of G it will be A. Okay, but the second allele is G. Okay, so there is no change in the second allele. So here we have in one allele we have this SNP with this reference number, but in the second allele we don't have. So we call this genotype, and here both of them, and again this is heterozygous. We call this heterozygous, and both of them, they are one, because one is different from the reference genome, and one, like, the reference genome. This one is homozygous, but there is a mutation in both of them. In both of the alleles, there is a mutation. All of them actually, they convert into A, but still it is homozygous. So as I say, sometimes these are associated, with a specific trait, sometimes specific with a specific disease. Some of them very severe diseases. Some of them may be just anonymous. That means they are not pathogenic. So this is the consequences of each of these mutation. There are different kind of consequences. Some of them benign. That means some of them likely benign. Some of them pathogenic. Definitely they will create disease or anything like that associated with disease. Some of them are likely pathogenic. Some of them are risk factors. So all these classification, we call them consequences of mutation in the annotation. And each one of these, they give information to the one who is, to the researcher. It's like if it's doctor, it will get some medical answer. If researcher also find, some uh, answer to some question and so on. So I think we're moving very slow, but I will stop right here.